Hello, my name is I양순교수. I will explain how laboratory tests for bacteria related to infectious disease, antibiotics, antimicrobial resistance, viruses, fungi, parasites, and others are being diagnosed in the Department of Laboratory Medicine. I currently work as an associate professor at Hanyang University Hospital. The Department of Laboratory Medicine is divided into 7 major fields: chemistry, hematology, transfusion medicine, immunology, clinical microbiology, molecular genetics, and informatics. Clinical microbiology is the field that identified and characterized pathogenic microorganisms, including bacteria, fungi, viruses, and parasites. In addition, antimicrobial susceptibility testing, AST, are conducted to assist patients receiving the proper antibiotics. Moreover, it is responsible for epidemiological investigation of outbreaks and hospital infection control. On the basis of the case, I will explain the diagnostic process. A 70-year-old male visited the ER with fever, cough, and sputum. Bacterial pneumonia was suspected on chest X-ray. Sputum culture and gram staining was performed. At this time, sputum culture samples and gram staining samples are referred to the Department of Laboratory Medicine sent to the lab. According to the established protocol, the following tests are performed in the lab to identify the causative bacteria. First, gram stain and bacteria culture are performed. If bacteria cannot be identified using a specific media or biochemical reactions, they can be identified by analyzing 16S RNA sequencing, which is their housekeeping gene, using PCR. Recently, the identification of bacteria using multi-top mass spectrometry has been performed very quickly and accurately. When bacteria are identified, AST are performed. The slides explain that in order to identify the causative agent of infection, we need to know the common bacterial pathogen associated with disease. The slide then provides a list of common bacterial pathogens for different diseases. This is schematic flow diagram of bacterial identification and AST conducted in the clinical microbiology laboratory. It starts by mentioning the different types of specimens we cast for culture in the lab, ranging from sterile to contaminated. This slide then explains how standard media is used for aseptic specimens, such as blood, body fluids, while specific media is used for contaminated specimens like feces. The slide also explains the role of gram staining and multi-top MS in identifying bacterial species and how susceptibility testing is conducted. And also the slide mentions typing, which is test used to identify factors that may have contributed to a bacterial outbreak. After suspecting the causative agent, the next step is collect the appropriate samples from the patient for laboratory testing. For example, in the case of suspected pneumonia, sputum samples can be collected and sent to the lab. The most critical simple test for predicting and identifying bacteria is the gram stain, which is a simple and basic test. This schematic diagram of gram staining process is shown. There is a basic difference in the cell wall of gram-positive and gram-negative bacteria. In the gram-positive bacteria, the peptidoglycan has a thick mesh-like cell wall, whereas gram-negative bacteria has a relatively thin wall with an additional lipid layer outside here, the outer membrane. The colonies are placed on the glass slide, fixed with heat, and then stained with crystal violet as first dye. The thick peptidoglycan in gram-positive bacteria is colored purple here, and gram-negative bacteria also colored. The third step involves the iodine fixation. The alcohol washout process is where the difference occurs. The thick peptidoglycan in gram-positive bacteria makes washout difficult, but the gram-negative bacteria with the lipid layer washout is nearly complete. Using safranin as a second counterstain show the gram-negative bacteria remain purple, but due to the failure of the washouts, while gram-negative bacteria are dyed red. After categorizing bacteria based on their gram stain, the next step is to examine their shape. Cocci and round and can be found in the cluster, chain, tetrad, or tetrads and pairs. For example, S. aureus appears clustered, while S. pneumoniae is mainly observed in pairs. If bacteria are not cocci, they are likely to be bacilli, like this. E. coli appears as a rod-like shape, whereas Clostridium has a box-like appearance. Coccobacilli are somewhat round but flat, and Acinetobacter baumannii typically resemble this type. Fusobacterium is a fusiform bacilli with a long, pointed end. Spirochetes are another type of bacteria that have a spiral or corkscrew shape. In addition to bacteria, fungi can also be observed using staining techniques. For example, in the case of Candida, gram staining reveals the presence of a black-colored yeast form here. And a test called germ tube can be used to observe the forming of budding, like this. The morphology of Aspergillus and Penicillium can be observed using the lactophenol cotton blue stain. In the example case, commonly acquired pneumonia, bacterial pneumonia is suspected, and the sputum test reveals a pear-like gram-positive stain, cocci. It is a strong evidence that S. pneumoniae is a causative organism. As another example, in patients with suspected genital tract infection, Neisseria gonorrhea can be suspected if the cocci from gram-negative is seen in the neutrophil, like this. It's also us to predict the causative bacteria to some extended basis on the patient's history, even if the identification is not confirmed after culture the bacteria. Let's learn how to culture a patient's sputum samples that infected by gram-positive cocci. In the previous case, as pneumonia was suspected, when sputum is cultured on the blood agar, the following characteristics can be observed. Blood agar literally contains sheep blood. As a result, the red blood cell is broken and turns light green as shown below. As pneumonia is very moist if you look like closely. As pneumonia can now be strongly suspected based on the patient's history, gram stain, and culture. There are also biochemical tests can be easily performed when there is a bacterial colony. Catalysis testing is one of the most representative biochemical tests. If bacteria has catalyst, when hydrogen peroxide is sprayed, water is decomposed into oxygen by catalyst, and bubbles form a result of oxygen generated. If we place the bacteria on the top of hydrogen peroxide, it will produce bubble if it has catalyst. If it is not, it will last as pneumonia does not produce bubbles, whereas S. aureus does. Differentiated which coagulates positive staphylococcus and coagulate negative staphylococcus is important. Bacteriological coagulase testing can be used using serum in the tubes. Coagulation occurs as follows if bacteria produce coagulates. However, bacteria lacking coagulates do not coagulate lacking or flowing. These panels produced by various companies can be used for bacterial identification using biochemical reactions. It can be used to differentiate between bacterial bases on how they respond to different substrates that are placed on the panel. A useful test for gram-negative bacterial biochemical reaction is triple sugar iron agar. These mediums. It is shown a ratio to 1 to 10 to 10 is used for glucose, lactose, and sucrose. And an indicator is also added to confirm the reaction, like phenolates. The response of each bacteria and type of sugar is breaks down to allow us to predict the bacteria. Bacteria can be either aerobic or anaerobic, and depending on whether they can survive in an environment with oxygen or not, they can be categorized into 4 groups, like A to E. Facultative anaerobes can grow in both aerobes and anaerobic environments. Aggregate anaerobes cannot grow in the presence of oxygen. Only the presence of oxygen allows obligated aerobes to grow. Microaerophils can only grow in the presence of a very small amount of oxygen. If you look at E tube, you will see that it is an obligate aerobe that can only grow in the presence of oxygen. B, on the other hand, is the obligate anaerobe appearance. D is facultative anaerobe, but while A and C are microaerophils. Abscess or pulse can be anaerobic infections. This is an aerobic chamber where anaerobic bacteria can grow. The chamber is filled with mixture of 85% N2, 10% H2, and 5% CO2 without oxygen. And the temperature is set to 37 degrees Celsius. Equipment for chamber is built heavy. Utilizing the anaerobic jar, will make diagnosis a little easier. When a gas pack is put into such a jar, oxygen is absorbed and other mixed gas are ejected. By inserting a medium, a gas pack, and locking the lid, the device maintains an anaerobic environment. The prevalent bacteria can be found in the humans are listed in these tables. Aerobes with gram-positive cocci and gram-negative bacilli are important. Among anaerobic bacteria, bacteroides are the most common. Clostridium is another important bacteria of great interest. Milditop's MS has been developed in the last decade and allowed for faster diagnosis than before. Applying to metal plates on the colony that have grown on the medium, when a matrix is derived and placed on the device disassembles it with a laser and analyzes as it flies through the vacuum tube. The x-axis represents mass and the y-axis represents the peak detected based on the mass. Using a database, it is possible to determine what type of bacteria it is. The accuracy was low in the beginning, but the technology advanced, it became widely used. The pictures are Milditop MS, which is being used in our hospital laboratory. As in the previous slide, is a picture of a flying colony on the metal plate, dropping on a drop of a special matrix and analyzing them in the equipment. Let's look at some representative bacteria. This is gram-positive Clostridium tetanus. It is the shape of a club, and you can see the spore. These are Propionibacterium acne, which is gram-positive and looks like a butterfly form, short butterfly form, and the Clostridium perfringens, like this, looks like a box. If it is closed, gram-positive cocci is S. aureus. In the case of Snocardia, it is branch form, gram-positive bacilli. This is gram-negative cocci. In the neutrophil, a case of Niger gonorrhea looks like this. Next, let's learn about AST. There are 4 major methods for testing antimicrobial susceptibility. Agar dilution is the gold standard. According to that, we are using gross microdilution, disc diffusion, and e-test more conveniently. The first is the disc diffusion method. Bacteria are added to the specimen medium at the specific concentration. An antibiotic-coated disc is incubated for 18 to 25 hours. Because the antibiotics spread around the disc diffusion disc, the bacteria are killed in the area near the disc. The area where bacteria do not grow in the known as inhibition zone, and it is measured to determine whether it is susceptible, resistant, or intermediate and resistant. Zone of complete inhibition could the diameter of disc show no obvious visual growth as judged by the unaided eye. Ignore faint growth of tiny colonies that can be detected only with the magnifying lens at the edge of endo in a zone of inhibition. This is e-test. This is method of testing by placing antibiotics on a bacteria-coated medium using strips with low to high concentration of antibiotics. Read the minimum inhibition concentration after 1 to 2 days of incubation. You can read the concentration of bacteria inhibited as shown in the picture. Concentration, inhibition concentration. The third method is gross microdilution. Bacteria are instilled at a specific concentration after antibiotics are divided into twofold, 1 to 4, 8, 13, and 16, 32, range from low to high concentration. It grew at low antibiotics concentration, as green color, grow of bacteria, but it did not grow at 64. As a result, the minimum inhibitory concentration, MRC, is 64. Bacteria grows as shown in the below in the circle in the shape. It's grow. Antibiotics are coated in the increasing concentration. Bacteria are dipping on it and grown for about a day, and the MRC is determined based on whether or not the bacteria grow. In the case of clindamycin, it is assumed that MRC was higher than 32 because of all of them reached the highest concentration of 32. In the case of penicillin, it's grow, not grow. Does not grow here. So this is MRC value. It's even in the case of erythromycin, so this is MIC value. Because it grew all the way and did not grow here. The fourth method is agar dilution. Like gross microdilution, sterilize antibiotics by the concentration. Fill the plate with antibiotic-containing agar and pour. It is a method of adding organisms and observed whether or not they grow. The following is antimicrobial susceptibility test and its interpretation guidelines. There are CLSI guidelines from the Clinical and Laboratory Standard Institute from USA and the UCAST guidelines from the European Community. The guidelines are updated annually and are used for interpretation. For example, when testing staphylococcus aureus, this division can be used. Mullington agar media should be used at the time, and Mullington plus cation adjusted process should be used when microdilution. Method. The guidelines specify which medium to use, how much inoculum to use, and how long incubation for each test. As also QC management, you must ensure that examiner has proper tests with the QC strength. The American Culture Collection number should be used, and the guidelines recommended using SOS of corresponding number of disc diffusion and the following number of MIH testing. The interpretation of the results as follows. If oxacillin is tested against S. aureus, it should be based on cefoxetine 30 microgram disc. And if zone diameter is 22 millimeter work grade, it should be reported as susceptible. But if it is less than 21, it should be reported as resistant. I will explain some of key mechanisms that resistance develops. The most important type of gram-negative resistance is known as extended spectrum beta-lactamase, ESBL, which referred to enterobacterias that have enzymes that can be break down a diversity of beta-lactam antibiotics. Carbapenem is used for ESBL positive enterobacterial infection. As a result, carbapenem resistant enterobacterias appeared. Carbapenem resistance A. baumannii and carbapenem resistance P. aeruginosa are important. Common examples of ESBL including CTXM, TAM, SHV, JES et al. And carbapenemase gene including KPC, IMP, BIM, OXA, and NDM. Let's see the mechanisms of resistance in gram-negative bacteria and antibiotic effects. The resistance mechanism mentioned before was the mechanism by the enzyme beta-lactamase in the cell periplasmic space. There are several beta-lactamase. Furthermore, there is a hole in the cell wall called porin. There is a mechanism to reduce the number of porin to block antibiotics from entering. Also, an overexpression efflux pump mechanism that remove incoming antibiotics. When the antibiotics enter the enzyme system, the mechanism occurs that cause ribosomal mutations to prevent the staking or the causative target mutations. Ribosomal mutation, modification, antibiotic modification enzyme. Different resistance mechanisms are used to express antimicrobial resistance, but the mechanism represented by enzyme like ESBL carbapenemase are particularly potent and can be passed on by other bacteria. MRSA is the most prominent and important gram-positive cocci bacteria. The megagen produced PBP2A, which interfere with the binding of antibiotics. There are also vancomycin-resistant enterococcus and S. aureus. The mechanism of vancomycin-resistant enterococcus is to inhibit vancomycin binding to the cell walls by modifying the cell wall structure, like this. SPAN-A and BAN-B genes are associated with this mechanism. A chromogenic media is a special medium to use to detect antibiotic-resistant bacteria more easily and quickly. As shown in the figure, vancomycin-resistant E. enterococcus faecium colonies are purple, and the vancomycin-resistant E. fecalis appears green. So we can easily identify and interpret it. Double disk synergy readily place two disks at the predetermined interval. and observed how the two disks interact and how bacteria grow. If it is positive in double disk synergy, this bacterium has an enzyme that degrades antibiotics, so the space bulges like this between two disks. It is common use for the ESBL compound test. In the case of parasites, morphology is most important. A skilled expert examines the morphology under a microscope and diagnoses it. In the case of malaria, diagnosis is only possible if the morphology of the developing stage is well understood. As the stage of proliferate development are varied, wing stage, trophozoa stage, sticky joint, and gametocyte stage. Because P. falciparum has a variety of shapes, experts must know all of them to make a diagnosis. The same goes to vivax and also P. malariae. Additionally, it is possible to diagnose by observing age of eggs. When a fungus is suspected in patients, it is cultured in space medium, like Sabrodactylus agar. It might take a week, 2 weeks, or even 3 weeks because fungi grow relatively slowly. The morphology is used to make the diagnosis. Shape and colors. Lactopanel cotton wool staining can be used as a more detailed examination and diagnosis on the microscope. All of them are described as aspergillus, but you can see the flavus fumigatus has slightly different shape. And tereus is also different. So the final diagnosis is made by seeing the shape and color with the naked eye in the previous slides, and by combining the result of staining under a microscope. The following are virus diagnostic technique. Virus culture, detection of virus antigen antibody, and PCR of viral genomes are available methods. Virus culture can currently be done by shell bioculture, which is a method of growing a virus for about 5 days, and then fluorescence staining and observing it under fluorescence microscope. This method is not common use in the clinical setting because it takes a long time to confirm what type of virus it is. The basic ideas behind the antigen and antibody detection method is to attach the antibody, pass the specimen through the kit, and then once the antigen-antibody reaction occurs, display the color like this band, control band, and the adenovirus and rotavirus positive specimen. Viral PCR needed accurate information of the viral genome. The figure is showing a specific segment of SARS-CoV-2. It is necessary to find a segment of a genome with a high specificity and sensitivity and amplify that segment in order to diagnose the virus. When identifying a specific virus, the best segment of PCR and the appropriate technique such as multiplex PCR, real-time PCR, nested PCR, or reverse transcriptase PCR should be used. In fact, the clinical microbiology lab where I worked focused on the bacteria identification, susceptibility testing, and even resistance mechanism, while viral PCR is handled in the molecular genetics laboratory, and antigenic reactions is handled in immunology laboratory. These pictures are PCR equipment in our hospital labs and molecular genetics lab. This is nucleic acid extraction equipment. This is the real-time PCR, and it is being analyzed. There are also devices that perform the nucleic acid extraction and PCR at once, like this. Please send me an email if you have any question. Thank you for your attention.